module, I will discuss about the structures of DNA and RNA. The structural components of DNA and RNA are as follows. The components of DNA, that is deoxyribonucleic acid, is purine, that is it consists of adenine and guanine, and pyrimidine, it consists of cytosine and thymine, and the pentose sugar. In case of DNA, it is deoxyribose sugar. DNA is double-stranded. On the other hand, components of RNA, that is ribonucleic acid, it consists of purine, adenine and guanine, and pyrimidine, that is cytosine, and in case of RNA, thymine is replaced by uracil and a ribose sugar. Now pyrimidine, it consists of cytosine and thymine. So pyrimidine, it is a single ring structure, that is pyrimidine ring. In case of cytosine, it is a 4-amino 2-oxypyrimidine, that is in the 4 position one amino group is present, and 2-oxy in the 2 position one keto group is present. So it is 4-amino 2-oxypyrimidine. On the other hand, in thymine, it is 2,4-oxy-5-methylpyrimidine, that is in the 5th position one methyl group is present, and in the 4 and 2 position 2 keto groups are present. Now in purine, it consists of adenine and guanine. Purine, here there is a pyrimidine ring and an imidazole ring, that is 2 rings are there. One pyrimidine ring, it is binds with an imidazole ring. Now adenine, it is a 6-amino purine, that is in the 6th position one amino group is present. On the other hand, in case of guanine, it is a 2-amino, that is in the 2 position one amino group, and 6-oxypurine, that is in the 6th position one purine group is present, that is one keto group is there on the 6th position. Now here in both case of pyrimidine and purine, these tautomeриc shifts are occur, that is keto forms are changed to enol form, or amino group is shift to imino group. These are called tautomeриc shifts, which are also occur in the case of these nitrogenous bases. Now in case of RNA, which is a single-stranded structure, here instead of thymine, uracil is present. So there is a difference between thymine and uracil. That is uracil, it is a 2,4-oxypyrimidine, that is in the 4th position, and in 2 position 2 keto groups are present, while 5th position methyl group is absent. So this methyl group is present in case of thymine. So this is the difference between thymine and uracil. Moreover, there is also difference between DNA and RNA in sugar. In case of DNA, 2-deoxyribose sugar, that is in the 2' position there is no hydroxyl group in the 2' position. On the other hand, in case of ribose sugar, in the 2' position one hydroxyl group is present. So this is the difference between DNA and RNA. RNA is single-stranded and ribose sugar is present, and instead of thymine, uracil is present. On the other hand, DNA, it is a double-stranded structure. Now, DNA, its building units are nucleosides and nucleotides. So nucleosides, it consists of a base and a sugar. So deoxythymidine means that is the thymine base binds with a deoxyribose sugar. So it is called 2-deoxythymidine. On the other hand, in case of your adenosine, it is also a nucleoside. So here, when a ribose sugar is bind with an adenine, it is called adenosine, which is a nucleoside. Nucleotides, which is a building blocks of DNA molecule, it consists of a base, a sugar molecule, and a phosphate. So in case of DNA, that is a base, suppose a thymine, it is binds with a phosphate molecule and a sugar molecules. That is a base binds with sugar and a phosphate. So it is a deoxythymidine monophosphate, DTMP, deoxythymidine monophosphate. So it is a pyrimidine nucleotide. Another purine nucleotide, it is deoxyguanine monophosphate. So deoxyguanosine monophosphate or DGMP is a purine nucleotide where a base, that is guanine, attached with a phosphate, with a sugar and a phosphate. It is first attached with a sugar, then with a sugar is attached with a phosphate. So it is a nucleotides. Now double helix, the structure which was proposed by Watson and Crick, is based on two evidences. One of the most important evidence is provided by Chargaff, that is the chemical analysis. According to this chemical analysis, the sum of purine is equal to the sum of pyrimidines. Then the sum of amino bases, that is adenine plus cytosine, is equal to the sum of the bases guanine plus thymine, and A is always pairs with T and G with C. So they are always present in equimolar amounts, that is AT and GC. So these are some chemical evidence provided by Chargaff. Moreover, another important evidence based on which double helix structure was proposed, it is the X-ray diffraction studies or X-ray diffraction data on DNA, which was provided by Wilkins and Franklin. So based on these two evidences, Watson and Crick proposed double helical structure of DNA. Now in the double helical structures, the sugar phosphate backbone is there. So bases are attached with each other by hydrogen bonding. Hydrogen bonding is there. Suppose in this figure, so GC, guanine and cytosine are connected by 3 hydrogen bonds, whether adenine and thymine is connected by 2 hydrogen bonds. The strands are antipolar, that is 5 prime, 3 prime, and that is antipolar. They are in opposite polarity. Now double helical structure, which was proposed by Watson and Crick, here the 2 strands are complementary. So here one red color and one is a green color, 2 strands which are complementary. One is 5 prime 3 prime, another is 3 prime 5 prime. The bases ATGC are stacked on each other, and they are at a distance of 3.4 angstrom apart or 0.34 nanometer apart. The helix turn at every 10 base pair, that is at 3.4 nanometer or 34 angstrom, the base rotates at 360 degree, and this turn of the base in each turn of the base, there are two groups are formed. One is called major group, another is minor group. Minor group is the smaller one, while a major group is the bigger one. The diameter of the DNA is 2 nanometer or 20 angstrom. So this is the one of the double helical structure which was proposed by Watson and Crick. Now this double helical structure which was proposed by Watson-Crick, it is the B form of the DNA, that is here helix are right-handed. So other forms are also there which we will discuss elsewhere. So in the conclusion, we want to say that is the discovery made by Watson, Crick, Maurice Wilkins and Rosalind Franklin, that is the discovery of the structure of DNA, it changed is one of the important milestone in science of biology, and it also changed the research and enhance our knowledge in the science of genetics as well as in biology. Watson and Crick and Wilkins, they received Nobel Prize in the year 1962 for this discovery, while unfortunately Rosalind Franklin, she died at the premature age of 37 years, so she could not receive the Nobel Prize, but she has also got an equal contributions in the discovery of DNA double helix. In this module, we have provided also key concepts and summary, list of books for your reading and study materials which you can download. Moreover, if you have any question, please feel free to ask us. Thank you for participation in our online course.